Hello CSIR JRF aspirants. Today we are going to learn about apoptosis. Okay, so we are going to see apoptosis in human and apoptosis in C. elegans. So apoptosis, why it happens? It does happen because there is the cell death, or apoptosis is basically a cell death, or whenever the body there is the two process, one is cell death and cell survival. Due for that, there is must, if there is senescence happens, means aging happens in the body, the apoptosis is required. Basically apoptosis is good for health, good for body. So the other term is very similar to apoptosis, is or very comparative to apoptosis is necrosis. So let's see a few points about necrosis and apoptosis. Okay, we'll see what is different, what is the difference between both of them. First is apoptosis. So apoptosis is basically single cell phenomena, only single cell are going to get die, and in necrosis there is a sheet of cells involved, because necrosis happened due to the accident, injury, etc. In the apoptosis there is no inflammation and bursting, but in necrosis there is inflammation and bursting happens. Okay, and I think you can see more clearly now. Yeah, third point is energy dependent process, and energy dependent process, and the in this necrosis energy independent process. Fourth point is regulate. Apoptosis is regulated and reversible in nature. It means it gets revert whenever the body required that thing, they are going to make it again happen. It is uncontrolled and irreversible. In this, plasma membrane remain intact. In this, plasma membrane are breakdown. No role of cytosolic organelles except mitochondria, and this role of cytosolic organelles such as rupturing of mitochondria, disaggregation of ribosome, disruption of endoplasmic reticulum. So these are a small difference between apoptosis and necrosis. Now we'll see the detailed process of apoptosis. So basically apoptosis occurred by two pathway. First is an extrinsic pathway and second one is intrinsic pathway. Extrinsic pathway means the pathway which is activated due to the extrinsic factors. Okay, so after that there is activation of disc, that is death induced silencing complex, and then the cell death will going to happen. And for this extrinsic pathway, the two important cellular signal participating in cell death and cell survival are we'll discuss it later. Okay, now the second pathway is intrinsic pathway, means it is means that is there is this internal signal that is due to stress. If the calcium concentration increase or injury happens, then the intrinsic pathway will occur, and because of that a process happen, and in the between of that there is a release of cytochrome C from mitochondria. Remember, if in the mitochondria there is cytochrome C. Okay, so if cytochrome C is get out of it, it means the apoptosis are going to happen. Okay, so what cytochrome C do? It will going to get bind with APF1, and then it will going to activate CASP9 and CASP3. CASP is caspase. Okay, so basically there is two type of caspase. First is initiator caspase, and second one is effector caspase. Initiator caspase are caspase 8 and caspase 9, and effector caspase are caspase 3 and caspase 7. So basically what is caspase? If I'm talking about, caspase stands for basically this enzyme contains cysteine rich at active site, like this. Okay, so there is cysteine in between of the active site. Okay, and AS stand for aspartate residue, and A is whenever A is there is a protease that break the peptide bond which involved aspartate. Okay, so basically this is the caspase protein that we are going to see in a more detailed way by this pathway. Okay, so first we are going to see the extrinsic pathway. As you can see, in the extrinsic pathway, basically if there is any due to the any extrinsic factor. So basically in the plasma membrane there are there is a presence of FAS receptor or TNF receptor, okay. And in basically what is FAS receptor? So basically, FAS receptor is a transmembrane type 2 transmembrane protein, and it belongs to the tumor necrosis factor. Okay, so there is the presence of receptor on the cell, and the ligand are going to bind, and there is a presence of two death domain. Death domain are going to activate just as similar to TNF receptor, tumor necrosis factor. They're going to activate the other two, that is TRADD, that is TNF, that is tumor necrosis factor receptor associated death domain, and the FAS receptor associated domain. Okay, after activation these two factors, it will going to activate initiated caspase that I am already discussed, that is caspase 8. And there are few protein that I already discussed in the earlier video, which is the pro-apoptotic. One of them is bid protein. It will going to activate the bid protein. How it will going to activate it? It will going simply, you know, cut the small part of the bid protein, which is the, I guess, C terminal, and the bid protein is get activated, and it will going to do or create a pore in mitochondria membrane, and then because of that cytochrome C will going to come out, and it will going to activate C3, which is caspase 3, and apoptosis will going to happen. So this is the small process, and same with the FAS ligand or FAS receptor. Okay, so let's see few pro-apoptotic factor and anti-apoptotic factor, so that it will going to help you in the next few more topics, okay. So yeah, pro-apoptotic factor. So here I am listing out. You just remember, because it will going to help you in the exam. So bad, bags, bid, back, big, noxa, puma, they all are pro-apoptotic. It sounds like bad, you know, B, and there is NP, that is noxa and puma. In anti-apoptotic, it inhibit anti-apoptotic means it will going to inhibit apoptosis, that is BCL2, BCLX2, BCLXLS, BCLW, NFA, and A1, that is annexin 1. Okay, so these are the pro-apoptotic factor, which, you know, do these factors are helping apoptosis, and anti means it will going to inhibit. Okay, let's see the we are going to now see the next pathway, that is the intrinsic pathway. Okay, now as we already seen why the intrinsic pathway happens. So normally in the cell, if the DNA is double stranded, right, there is a p53 gene, that is a tumor suppressor gene. It will normally it is present in the cell with MDM2, and there is the ubiquitin attached. Whenever ubiquitin is there, there is a degradation happen. It means no role. If any injury or internal injury happen, or internal signals happen, the DNA is breakdown. Now after DNA is breakdown, there is two protein arrive there, that is ATM and ATR, and then it will going to activate check 1 and check 2. Check 1, check 2, what will they going to? They're going to phosphorylate p53. They are going to phosphorylate MDM2, and MDM2 are going to detached from p53, and p53 get activate, and it will going to activate all the pro-apoptotic factor, and apoptosis happen. How? Same as we can see in extrinsic, they are going to activate bad and back. Okay, and what they are going to, they create a small pore in the mitochondria, and cytochrome C will going to come outside. Here the one more thing is happen. The cytochrome C are going to bind at APAF, which is APAF1, are going to bind at C terminal. There is two terminals, C and N terminal. After binding this cytochrome C and at this C terminal, there is the second thing happen. The ATP binding site will going to create, and there is the requirement ATP. That is, we already seen apoptosis is a ATP demanding process. It requires energy. It is an energy dependent process. Then APAF are going to activate, and it will going to bind at initiator caspase, and this whole process happens at 7 times, means 7 APF are going to bind at caspase 9, and it forms apoptosome. Okay, then after activation of caspase 9, that is initiated, it will going to activate effector caspase, that is C3 and C7, and then the apoptosis are going to happen. So this is the intrinsic pathway. I hope you will understand. So two important cellular signaling participating in cell death and cell survival are TNF alpha signaling. What TNF alpha signaling? Tumor necrosis alpha signaling happens. This signaling occur usually in extracellular apoptosis. TNF alpha is a pro-inflammatory molecule which interact with dimeric FAS receptor, as we already seen. Upon interaction with FAS receptor, it activates the caspase 8 protein. And caspase 8 downstream activate pro-apoptotic protein, that is BID protein. BID is responsible for promoting cell death. TNF alpha also responsible for inhibiting expression of IAFs. FAS receptor belong to death inducing silencing complex, that is DISC. So there is a small note that basically cytotoxic T lymphocyte. In the immunology, it usually expresses the FAS ligand, which interact with FAS receptor expressed on the target cell, which is going to that is going to destroy. Upon interaction, T cell causes cytotoxic mediated killing of the target cell. So this is how the cell signaling happen. Now the second signaling is PI3 kinase signaling. It is important for cell survival, and it is activated by growth factor and insulin. Okay. So today we are going to discuss different cases in our process. Okay. So suppose if there is ATP and caspase is not available, then the necrosis happens. If there is if the caspase 3 or caspase 9 are knockout, then malignancy happen or a cancer arise. If the APF are not going to bind, basically the inhibitor of APF is heat shock protein 98. So then only then also the cancer arise. So these are the situation where cancers arise, and these type of questions are usually asked in the exam. Okay. One more signaling is there, that is the decoy receptor signaling. They are inhibitory signaling receptor which interact with growth factor and cytokines, especially. For example, TNF-R6, it is a receptor of TNF alpha which inhibit apoptosis. So inhibitor of apoptosis is TNF-R6. IL-1R, interleukin 1, it is a pro-inflammatory molecule which promote the inflammation. Okay. When IL-1R bind to the decoy receptor, it inhibit the inflammation. Okay. I hope you learned something from this video. Happy learning. All the best.